I have incredible anxiety right now. No, but let's go.
All right, everyone, please welcome up to the front, Caleb Carter.
Hi, good morning, everyone. My name is Caleb Carter, and this is my senior presentation on the amplification of angiomotin expression through transient transfection in glioblastoma cells. Okay, so I'm going to give everyone just a quick overview of what my presentation will be about. Firstly, the cancer cell line that I did was U87MG, which is basically just glioblastoma cells, which is brain cancer. Now, the importance of studying brain cancer, you can either do trying to find like specific targets in cancer, or you can simply try to figure out different types of machineries that cancer, utilizes and has. The problem with cancer now is that, like we heard, the only treatments that we have for it is either surgery, chemotherapy, radiation therapy. And as we heard, these three, while it does kill cancer cells, it also deals a lot of damage to our normal healthy cells, which causes a lot of harm for people. Specifically, in the past, scientists wanted to find a difference. What is the difference between a cancer cell and a normal cell in terms of gene expression? And a specific gene that we found was one called AMOT or angiomotin, which I'll explain later. And so for my research, we wanted to target that specific gene. Okay. And so the goal of it is to elicit a overexpression of angiomotin and then see what correlations that has to other genes that deal with cancer activity. And how we did that through our experimental methods is through a transient transfection, which I will explain later, but for now, just know that it deals with short-term effects of an altered gene. And then we'll go into results, which, spoiler alert, it did work. We did overexpress angiomotin, and then it did have effects to the other types of targets that deal with cancer. We'll go into discussion, and then later we will talk about future directions that we can go after this experiment. So cancer. As we heard multiple times, cancer is an unregulated, uncontrolled cell growth. So essentially, your normal cells will eventually keep on proliferating and then create abnormal cells. These abnormal cells will then go to tissues, will go to organs, and do a lot of damage to it. And so how is it caused? There are many different factors that play into this, specifically things with the genes. You can have just a predisposition because of like your parents having it. But it's usually caused by some type of mutation, either from like environmental, smoking, things like that. Lifestyle also plays a huge role into it. And so what this does is creates problems in the cell cycle. So when one cell wants to go into two cells or like have mitosis, it has to pass a certain amount of checkpoints. And what happens is when these checkpoints are not regulated, it will start proliferating and creating more cells. And this goes from a normal cell into an abnormal cell because things like the chromosomes aren't lined up correctly, these things, and that is what causes cancer. Prognosis. This is kind of a broad idea because there's so many different types of cancers. In some types of cancers, you have a decent prognosis and everything. If you catch it early, it's like a low stage and not very aggressive cancer, you have a good prognosis. While others, it's entirely different and it's very aggressive. A specific example is brain cancer. Now, the specific cancer that I did was glioblastoma. There's a level of 27 different types of cancers that have a chart and the rate of occurrence on all those levels. So brain cancer was ranked 17th out of 27. So relatively on the low end, but enough to make it still relevant. However, when you compare that to how often does it have detrimental effects and life-threatening, it was actually ranked 9th. So it went from 17th all the way to 9th. So that's a huge jump comparative to how many people it is affected to how many people it's actually killing. And then for treatments for this, what would normally happen if someone had glioblastoma, you cut out, a portion of the skull, take it off, you cut out as much of the tumor as you can, but the problem is when you have a tumor in the brain, it's going to cause, since it's in places, it might be like inoperable. So you can't actually do anything to it. So after that, you're going to try things like chemotherapy, radiation therapy, just trying to lower the risk of it. A problem with specifically glioblastoma, though, is that even if you do get all the cancer or you shrink the cancer, has a very high chance of 95% of a reoccurrence. Also with glioblastoma, the rate of, your 5-year life expectancy is only around 7%. And a majority, the average of patients with glioblastoma only live up to 9 months. Now, what makes this so difficult to treat is a part of it is the blood-brain barrier. So our body has this natural barrier, and it's really good to block our blood cells from our brain and not bring like diseases and problems to the brain and cause problems. But it's really good for that sense. But once we have cancer, how are we going to treat it? We're going to have find something that can go through the blood-brain barrier and specifically circumvent that whole stopping that is specifically there to prevent things from entering the brain. So that creates problems for drugs. So let's say hypothetically a drug is created tomorrow. All cancer is cured. How are we going to get to the brain though? It's not as easy as some types of cancers because it doesn't have a blood-brain barrier, but this one does. Now the two types of cells that are in the brain is your neurons. And basically these are your things that elicit impulses. Basically anything you can think of, neurons are doing it. And then you have your neurons, and then you have all your supporting cells. You actually have 10 times more supporting cells than you have neurons, just proving how important neurons are. Your first type of glial cell is your oligodendrocyte. As you can see there, it says wraps myelin around multiple axons. All myelin does is pretty much speeds up impulses because we want things to be very quick in the brain. We don't want to be lagging behind. Your second one is microglia. This deals with things like immunity. If something does get through the blood-brain barrier, this is what's going to take care of it. But most importantly, astrocyte. These deal with pretty much everything from blood vessels to neurons. And the important thing about this is glioblastoma is also called level 4 astrocytoma. So these are the astrocytes, the cancer of the astrocytes, which is a very aggressive cancer. And the reason why it's so aggressive is because of angiogenesis. Now, what is angiogenesis, you might ask? It's literally the creation of more blood vessels. That's what it is. And naturally where your body has this is in embryonic development. And then normally, so when you're born and you're a kid, you need to grow, so then you're going to create more blood vessels to increase in size. Very simple. But once you're an adult, your body doesn't really use it unless you have a wound that you need to heal or in a woman's menstrual cycle. These are the only times that there's a spike in any type of angiogenesis. However, cancer hijacks this. It'll take over this whole thing and steal all the supplies for itself. Think of it like this. Your body is like a city, and your blood vessels are the streets in the city that bring in materials, okay? What cancer does is it steals that whole machinery to make roads for itself. So it just keeps on feeding itself. As you can see here, it will grow, grow, and grow. But specifically in glioblastoma, though, since it's a very aggressive, instead of making roads, it makes highways. That's why in reoccurrence, it's so, it can even when you get rid of all of it, it just reoccurs and it's stronger the second time. It's because it has such incredible angiogenesis. And so how does angiogenesis start is a specific gene called angiomotin or AMOT. And so the functions of AMOT is really the start and then the continuation of angiogenesis, and it does this through endothelial cells. So endothelial cells are the makeup of blood vessels. And then angiomotin deals with many targets that deal with the size of the organ and stuff. Specifically, yes, associated protein or YAP and connective tissue growth factor. So these two, YAP deals with growth size of organs, and then connective tissue growth factor deals with the migration of cells, the adhesion of cells, and the proliferation of cells, which is just continually making cells. And so you can see how if these three genes are dysregulated, this can cause the phenotype of cancer to persist. So in my experiment, firstly, the first thing that you have to do to even do anything to a cell line is you need to make a cell culture. And so for cell culture, and our specific is U87MG, so glioblastoma cells. So you get the cells as previously mentioned. A patient will give cells for us to study on, and then we need to create the exact environment, okay? Because the laboratory environment is very different from the human body, so we need to, monitor pH, temperature, oxygen levels so that we can mimic close enough to a brain chemistry as we can. Then we're going to a transient transfection. Now, this is a little confusing, but what it is essentially, it's like sending mail to someone, okay? So first you have your letter. And then this letter for this specific thing is DNA. So imagine a letter and the letter is DNA. And then you have an envelope that the letter will go into, and then we're going to place this specific, like the DNA with the envelope, we're going to place it on our cell line, okay? What that does is it basically just tricks the cell into expressing that type of DNA, because that DNA will go all the way from outside the cell all the way to the nucleus, and it will, the machinery of the cancer cells will read that DNA and then make it into RNA, which then goes to our next step of extracting that RNA. All this means is we're going to break open a cell, and then we're going to separate the different labels of materials that we have from a cell and just only take the RNA, because this is the expression level. Then we're going to go back into cDNA to place it into a quantitative polymerase chain reaction. This just quantifies or tells us how much of an expression that we had, and then how we can relate that to like a control. But specifically to understand this is like DNA. So when we first placed our letter in there that had DNA, we gave it to the cell. Here's DNA, make this. So it's going to transcribe this using enzymes into an RNA sequencing. And then when we took that out, we turned it back into reverse transcriptase. The importance of this is every part of your, every cell in your body has the exact same genome, has the exact same DNA. It literally only matters what is being transcribed to show the difference between two cells. And so for our results, we have angiomotin, AMOT, YAP, and CTGF at the bottom on your x-axis. And then our y-axis, we have our relative fold change. What this means is the difference between our control group and our experimental group. And our experimental group is shown here. Our control group is simply set to 1. It's an automatic set to 1. As you can see, angiomotin, we did very well in expressing angiomotin. It went from 1 to 386, so that's an incredible expression. But then YAP and CTGF both went down by 50%, which might seem confusing. So why did angiomotin, which targets YAP and CTGF, why didn't those also go up with it? And the reason why this is so angiomotin specifically binds to YAP, okay? And when the cell realizes that we've made way too much angiomotin, it needs those materials back. There's only a limited amount. So what it's going to do, it's going to create a drain, and it's going to literally recycle every part of angiomotin that it needs back. So when angiomotin starts getting recycled, since it's bind to YAP, YAP will then be lowered. That's why it's gone down by 50%. And then CTGF is a target of YAP. So when YAP goes down, CTGF goes down. So what future things can we do? When you deal with a gene and you want to know what a gene does, you can do one of two things. You can silence it or you can express it. And then you see what its targets, what happens to its targets. In this, we overexpressed it. So a brilliant idea would be like, let's silence it. What does that do to its targets? Also, in angiomotin, there's two different types of isoforms, AMOT 80 and AMOT 130. They do different things, and they have different activities with cancer. So knowing the differences would be a smart way for next research. And then angiomotin in normal brain cells to show, kind of mimic the idea that cancer has so that we can follow it step by step. And lastly, I would like to thank Dr. Ranahan. He's saved me multiple times on this presentation, in this whole research, and then the whole bio department definitely helped me. Dr. Gelders saved me emotionally through this whole thing. And then Diego, you heard me, and then Natalia has helped the whole transfection. Thank you. Any questions?
You mentioned transient transfection.
Yes.
Right? And you mentioned there's two ways to study a gene. One is to silence it.
Yes.
So let's say we wanted to overexpress it a different way.
So there is a transient transfection, which shows it here. But there's also like a stable expression. And what this means is you're going to put something on like a lentivirus, put CRISPR into the cell, and it's going to basically remove all the methyl groups so that it can be like transcribed. And what this does is instead of it going up from like 1 to 386, it'd be very stable. It's in the genome. It's a very slower approach. And it would, personally, I think it would be a great idea for future ideas because it would only go up so much, so then we could actually see what happens to YAP and CTGF. Because in that case, what I would predict would happen is angiomotin would go up, and then YAP and CTGF would also go up as well. Yes.
In your graph, on the y-axis, you have those two.
Okay, so the bars, is that what you're asking? Okay, so the bars. Basically, in a graph, it's impossible to put the number 386 right next to 0.5. Visually, it is literally, you can't see. I literally, when you make a distance to Excel and you put 386, it literally, there's just one bar. So it's like the only reason I have to create the break, it just shows that it's continual expression all the way up to 386. That's why there's the breaks on the side.
Thank you very much.
Cheers.
Okay, so we are, now we're going to take a break. We'll have a lunch session, and then we would ask everyone to come back around at noon for the plenary speech.